Hello everyone. Today we are going to learn about fungal staining. Okay. So in this picture you can see this is the type of fungus. This is one of the fungus that is grown on the plate, and this is the mushrooms. Okay. So all this type of fungus we are going to learn how to identify this fungus using staining method. Okay. So, fungal staining method is a simple histological staining method that is used for microscopic examination and identification of fungi. Okay, this is also a histological staining method and a differential staining method. Okay, so different types of staining method we have learned in our previous video, in our staining video. If you haven't watched it, you can go and check it out. So from there we have understood that the differential staining procedures or the methods, what they do, they identify the structures. Based on the structure, they can identify what type of microorganisms they are. Okay, so this is the this fungal staining method is a type of the differential staining method. Now, as we know, fungus are eukaryotic organisms. Okay, and they have both microscopic and macroscopic character. Like macroscopic means like mushroom. Okay, so they are large and they can be grown outside, and the microscopic characters which can be grown in the plate and you need the microscope to observe them. Okay, so these fungi are have both the characteristics, right? Now this fungus, what they have propagules, and these propagules are either hyaline, that is colorless, or they have different colors. They can occur in different colors also. Now these fungal spores, these spores are made up of chitin. Okay, and the hyaline mycelia spores and conidia, what they do, their cytoplasm, they can be stained using the stain, that is fungal staining method. The stain that we used for the fungal staining is the lactophenol cotton blue. Okay, so this is a very widely used and commonly used staining solution for this fungal staining. Now, this lactophenol cotton blue solution acts as a mounting solution as well as a staining agent. Okay, and this cotton blue from this solution, it stains the cytoplasm, which results in light blue background. Okay, so lactophenol cotton blue stain is a clear and a blue color solution. Clear means what? It is completely dissolved. Okay, and it has blue color, and it is basically made up of a combination of the three main reagents, that are phenol, lactic acid, and cotton blue. Okay, so all the three reagents have different types of functions to do. That is, phenol is acts as a disinfectant by killing any living organisms. Lactic acid, it preserves the fungal structure from being distorted, and the cotton blue gives the color to the chitin that is present on the cell wall and also gives color to the other fungal structures. Okay, so if we summarize it, we'll find that lactophenol acts as a cleaning agent, whereas phenol as fungicide. Now this lactophenol has come from mixing two reagents, that is lactic acid and phenol. So from there the lactophenol has come, and the third one, that is cotton blue, is gives its complete name, that is lactophenol cotton blue stain. Okay, so this fungus are basically very commonly grown, you can see commonly anywhere, and they do not require any particular conditions to grow. Okay, so they, not all fungus are pathogenic or harmful to humans, but some fungus are pathogenic to humans and to other animals also. Okay, so they also come under the notorious types of microorganism, and the structures you might have seen in your plates growing. Okay, so you have molds, you have yeast, mushrooms. Some fungus are also edible, like mushrooms, like yeast. Okay, which is used in bread making, in bakery. Okay, so all this fungus, they are not they do not require any particular growing conditions, right? So they are the common contaminants that we can find in our microbiology lab. Okay, so coming to the requirements for this staining, we need a young fungal cultures. that should be 5 to 7 days old only. You need to have young for proper identification, young cultures. Then we need a mounting fluid, that is lactophenol cotton blue solution. Then we need glass slides, cover slips, needles or inoculating lube. You can use ethanol 95%, UP roll or nail polish, and microscope. Okay, so here a UP roll or nail polish is basically used in case of a permanent slide. Okay, so if you want to make a permanent slide of the fungus, then only you will use the UP roll or nail polish. Otherwise, commonly it is not used for regular practical purpose. Okay, so these are the requirements. Now coming to the preparation of this cotton blue solutions. Now what are the requirements for making of this solution? These are distilled water, cotton blue, phenol crystals, glycerol, lactic acid and 70% ethanol. Now there's something that we should know, that is lactophenol cotton blue solution is always prepared at least 2 days before. Okay, it is to be made fresh every time you do the fungal staining, and you should make it before 2 days to allow dissolving and maturation. Okay, so coming to the preparation process of the lactophenol cotton blue. Now in the day 1, what you do, you dissolve the cotton blue in the distilled water and leave it to rest for overnight. You keep it there. This will eliminate the insoluble dye. The cotton blue will completely dissolve in the water and all the insoluble components will be eliminated. Now in the day 2, what you do, you take a glass beaker, put a lactic acid in it, and to the lactic acid add some phenol crystals to it. Okay, now everything, all these procedures should be carried out using protective gloves only, to avoid contact with your skin, because you are using acid. Okay, so use protective gloves for this making preparation of this lactophenol cotton blue solutions. Okay, now after adding these crystals to the lactic acid, what you do, you stir it using a magnetic stirrer until the crystal dissolves completely. Okay, you have to keep on mixing it until it is properly mixed. Okay, now after that, after mixing it properly, you add glycerol to it. Okay, proper amount of glycerol you need to add to it. Then filter the cotton blue and the distilled water into the phenol plus glycerol plus lactic acid solution and mix it. That means what you do, you filter the cotton blue solution that you have made in the day 1, you filter it to the phenol glycerol lactic acid solution that you have made in day 2, and after filtering it, after putting it, after mixing the two, you need to mix it properly. Okay, after adding the two of the solutions together, you need to mix it properly until it is completely mixed together, right? And then after that, you store at room temperature. Now the results will look somewhat like this. This is lactophenol cotton blue solutions. Okay, so this is how the cotton blue solution is made before 2 days. Okay, now after making, we'll come to the procedure. Okay, so in the procedure we have, in step 1, you take a clean glass slide and add a drop of 70% ethanol to it at the center. Put a ethanol drop at the center of the slide, and after putting it, in step 2, you add some fungal specimen to it. Okay, you take a fungal petri plate that you have cultured in your lab. Okay, you take a inoculating loop, take the culture and put it on the 70% ethanol that is you have which you have put in the middle of the slide. Okay, now remember, you need to take young cultures only that are 5 to 7 days old from the culture medium. Okay, now in step 3, what you do, you tease the fungal sample in the alcohol properly using a needle mounter or inoculating loop. Okay, so you need to tease it completely in the ethanol for proper mixing. Now add 1 to 2 drops of lactophenol cotton blue solution to the mixture using a dropper or pipette. Okay, so 1 to 2 drops is added, and you need to do it before the ethanol dries off from the slide. Okay, so ethanol is a volatile solution, it will dry up quickly. So before it dries up and after teasing it completely with the ethanol, you need to add 1 to 2 drops of the stain. Now carefully cover the stain with a clean sterile cover slip without making air bubbles to the stain. Okay, so here you need to remember that you have to avoid making air bubbles on the slide. Okay, as this will interfere with the clear visualization of the structure, right? So air bubbles should be avoided at any cost. And lastly, step 7, you have to observe it under 40x and then you observe it under oil immersion objective of the microscope. Okay, so this is the complete procedure. This will become more clear if you look into the pictorial procedure of the staining. So in step 1 we had ethanol. A drop of ethanol was placed on the middle of the slide. Then we had taken a petri plate which has a fungus culture. Fungal culture, we have taken an inoculating loop, take the culture from it, and what you do, you place it on the middle of the slide where you have put the ethanol, right? Now in step 4, you do the teasing part. Tease it properly with the 70% ethanol for clear visualization. Then you add 1 to 2 drops of lactophenol cotton blue to the mixture. Okay, and after that you do put a cover slide on the smear on the teasing fungal specimen which you have teased. Okay, you put a cover slip on it and you look it under the microscope, you observe it under the microscope, right? So this is the complete pictorial representation of the complete process. Okay, now what you will see under the microscope if you put the slide. So you will found that the fungal spores, hyphae and the fruiting structure have stained blue color while the background has stained pale blue color. Okay, so the spores, hyphae and fruiting structure will somewhat look very dense blue and the background will look as a pale blue, right? So this is a image of a fungus and this is the original microscopic image. So this is the image of Trichophyton mentagrophytes and these are the fungal hyphae with spores. These are hyphae that have spores above it. So here you can see that the hyphae and the spores have taken the dense blue color of the solution while the background is taken a pale blue color. Okay, now if we look at another example, here you can see this is another type of fungus and this is the microscopic image. This is the example of Aspergillus niger and these are the fungal spores. Okay, now you can see here that the spores have taken the dense blue color while the background has taken pale blue color. Okay, now the structures are quite different from each other. Now since fungus are present in a wide variety, okay, they have variety of structures, so different types of fungus are present. So we'll not go deep into that. If you want a separate video on the types of the fungus, you can put your suggestion in the comment below. Okay, so we can do it in another video. So in this video we'll focus on our staining part. Okay, so here the different types of fungus, you can see different structures is visualized under the microscope where the fungal structures have taken up the dense blue color of the lactophenol cotton blue solutions, whereas the background has taken the pale blue color. Now coming to the mechanism of the fungal staining. Okay, so what is actually making the fungus structure to stain in such a dense blue color? Okay, so if we look into this structure of the fungal cell wall, here we can see that it is made up of wide variety of components. Okay, so here in the upper portion we have the glycoproteins, that is polysaccharides and glycoproteins all are present, manoproteins are present, glucan is present, and below the glucan there is chitin and then there is plasma membrane. sorry, this is the peptidoglycan, this is the lipid bilayer, this is the peptidoglycan structure, and below that it is the cytoplasm. Okay, so this is the complete structure of the fungal cell wall. Now this cell wall is a characteristic of fungi and is composed of mainly glucans, chitins and glycoproteins. Okay, so as we have seen, this is the glycoprotein above, then below is glucan, then there is a chitin. Now this fungal cell wall is located outside the plasma membrane, uh, and it mediates all the relationship of the cell with the environment. It protects the contents of the cells, it gives it rigidity and defines the cellular structure as well. Okay. Now this lactophenol cotton blue solution, what it does, the lactophenol, it stains the glucan and the chitin components, while the cotton blue, it stains the cytoplasm. Okay, so lactophenol stains the glucan and chitin components, and these are they have a very um, strong network together. That is why they have taken up the lactophenol cotton blue solution, and they appear dense blue in color, whereas the cotton blue stains the cytoplasm here below it. And since the network is very loose in the cytoplasm, it's a fluid structure, so it will give a pale blue color. Okay, so this is how the stain um, gives a different colors at different densities. Okay, so as I have said, the fungal cell wall are not present in humans. Okay, these are also they also come under the most harmful and pathogenic microorganisms. Some of them are very pathogenic, and therefore this structure becomes an excellent target for antifungal therapy. Okay, so if we look for some the examples of the fungus, we have Trichophyton mentagrophytes, the example of which we have seen our previous one. Aspergillus niger, Penicillium notatum, very common fungus. Saccharomyces cerevisiae is yeast, we all must have heard about it. Cryptococcus neoformans and Candida albicans. Okay, now this Candida albicans is a pathogenic microorganism, a very mostly known for its infectious structure. For fungal infections, you can see. You must have heard about it, fungal infections. So, Candida albicans is the cause of all that infections. Okay, so this not all fungus are pathogenic, but some are, and they are very common contaminant that we face in our microbiology laboratories. Okay, even in pharmaceutical industries also. Okay, so we need to avoid this fungal contamination. So this is all for today's video. Hope you have understood this whole fungal staining procedure. If you have any queries related to the video, please comment it below. And if you want a detailed notes of this video, follow our LinkedIn page. The link is in the description. If you have liked this video, give it a thumbs up. And do share and subscribe to our channel for more such related microbiology updates.